doing her work in Paris. So she was studying, let's see if I get this right, Parkinson's disease, right? Okay. And then she eventually switched, hopped over the pond, came to Boston, and now she's looking at how hormones affect our behavior in humans. So it's really exciting, and this is the cool part, right? We get to actually meet someone who's doing things on the cutting edge of science. And that means that as you listen to her talk, I want you to collect questions and see so that at the end of the show, we can ask her all of our fun questions. But regardless, I want to thank Dr. Piet for joining us and hand it over to her. All right, here we go. Let's give her a round of applause.
Thank you. Thank you everyone for coming, and thank you also for the very kind introduction. And so yes, I'd like to share with you 3 kind of short stories that have been important for my little neuroscientific journey since I started. So as my PhD program in Paris, so I worked on actually 2 different projects. So the brain simulation and a big therapy right now for treating Parkinson's disease, but we don't really know why it's actually working. Also on the behavior side, I wanted to learn more with one-shot learning. So think about how a single, very brief experience can still trigger long-lasting memory and can transform memories very rapidly. And then moving closer in time, to what I'm doing right now is as you said, how hormones can impact our daily behavior. So I kind of tried to add into all of those 3 quite independent topics, also to emphasize to you that the journey of junior scientists can also allow you to have this freedom of choosing very different topics on which you can actually work quite in depth. And so to start with the first topic, I want to start with the learning one-shot memory aspect. So think about how learning, forming memories, and forgetting are really integral parts of your daily life, and how they can also have both positive, negative impacts. And what is quite intriguing is that we can form memories very slowly. Think of how, for example, you learn to ride a bike or play a musical instrument. But we can also form memories very rapidly. Think, for example, of how a kid can learn hundreds of words within a few weeks at a single kind of time period, or how if you were to meet your favorite actor, for example, in the elevator, probably this would trigger a long-lasting memory for the lifetime, right? So I wanted to kind of understand how this is kind of possible in the brain, and we don't really have a very clear answer of the kind of changes that are happening and that can underlie this type of learning of one-shot experiences. And so to study that in the lab, we kind of had to find a novel paradigm that we could use in mice, basically. And so which is kind of the animal model that we have to study the neural changes actually happening. And so what we kind of came with is this kind of sticky tape avoidance test. So you can try it on your cats or dogs, and it actually also works. In humans, probably less the case. Recently tried with a young kid, and they didn't really work that well. But so the idea is that we place a mouse in an arena, and there's this kind of little sticky tape that it can explore and then touch it. And it's kind of slightly uncomfortable, and so of course the mouse will try to remove it, and this actually happens very quickly, which is a nice thing because we want to study one-shot learning, so brief experience again. And so in our case, most of the mice remove the tape very rapidly, and what is quite striking is that if you look at the trajectories on the next day when we test the memory again, while the mouse is actually where is it? Avoiding the little tape that is here in green, right? So strong memory form that can last actually for a week and months even. And that was kind of our starting point to study this one-shot learning aspect in the brain. And we actually had like a major hypothesis in the field is that memories are kind of stored in what we call synapses, which are those kind of junction between neurons. So here's one synapse that is represented here, and what's quite nice about the synapses is that they can be relatively plastic. So the strength of the information flowing from one neuron to the next can be adapted, increase, decrease, depending on how much the neurons communicate between one another. And in our case, we had also one particular candidate for how one-shot learning more specifically could trigger long-lasting memories. And this candidate is actually this endocannabinoid system. So it's probably a kind of new word. But what is quite nice is that those endocannabinoids are actually very close to what people can use, as recreational drugs like CBD or like therapeutic drugs like CBD or THC. So they bind to the same receptors. And so the CB1 receptor or CB2 receptors, and so those receptors are initially meant to actually bind to not the artificial synthetic compounds, but to those endocannabinoid molecules that are endogenously synthesized by our bodies and also by the synapses. And so what we tried to see is whether or not by inhibiting those receptors, we could actually impair the learning of these one-shot experiences. And so we tried this hypothesis. So what you see here is some injection that are meant to delete the expression of the receptor in specific brain regions. So here on the kind of histology tissue pictures. We checked that it actually could prevent the induction of the synaptic plasticity in the brain. And what we found is that when we looked at the behavior of those mice, when we tested them in the arena, is that actually they were indeed no longer avoiding the tape. So you see the knockout, which no longer express the receptor, don't avoid the tape as much. And you see it by looking, for example, at the number of detours or the latency before they contact the tape again in the test session. So that's kind of one little story. And what I want you to also keep in mind is that we can now try to bridge different scales with the tools that we have in neuroscience now, from the behavioral kind of level down to the molecular, endocannabinoid in this case level. And this can also be very useful if we think of drug development, especially in the case of memory loss or memory improvement. So that's kind of the nice thing about this type of work, I would say. And then moving even more towards the kind of clinical side. So here's a second little story, which focuses on deep brain stimulation. So deep brain stimulation is a very efficient therapy that has been used especially in Parkinson's disease, and now has also been developed in other types of disorders, in also depression, for example. And so Parkinson's disease in the US is quite prevalent. You have at least more than 1 million patients that have been diagnosed, and 100,000 new patients every year. And it's chronic degenerative disease. But what people don't necessarily know is that it actually affects also young people. So 20% of the patients are actually below 50 years old. So it's very you have very strong motor symptoms like slowness of movement, tremors, also some cognitive decline. And what happened is that usually the classical treatment would be medication. So people would replace one chemical that is progressively lost during the as the disease progresses, which is dopamine. But at some point, this medication is no longer so much effective, and one option in that case is to implant the patient with an electrode, so deep inside the brain, and so that's what you can see on that CT scan. And so this is what we call deep brain stimulation. So the electrode is continuously kind of stimulating that deep brain area, and we don't really know why, but it works extremely efficiently. So if I kind of shows you this video, the patient was off just before, and now the stimulation can be turned on remotely, and you will see that the tremors actually suddenly almost instantaneously disappear. And not only the tremors, but also the walking and gait pattern improves very rapidly. So very efficient symptomatic treatment, but we don't really know how it works. So that's kind of the mysterious thing that I wanted to also understand a bit more, basically. And so for that, I look at it first in a very kind of theoretical, abstract way. And so to give you a sense of what I've been working on is that one important manifestation of the neuronal activity in Parkinson's disease, that neurons tend to fire kind of together, like in a synchronous way. And so creating those big waves of activity that kind of prevent information to actually like really flow more naturally. And so to study this synchronization aspect of the neural activity, we used mathematical equations, like models of neurons. And here is a kind of small video that shows you what we can do. And so each dot here will represent one neuron that is like again modeled by some mathematical equation. And what you see that, you know, in this initial region at the bottom, it's kind of the resting state, let's say, of the neurons. But what you also notice is that even though they have like the little beehive kind of colony, some neurons are a little bit more explorated and kind of cross this red kind of barrier. And as they do so, well, they kind of pull everyone together with them, and that creates this like little shift of activity. And so this is what we call like this kind of burst or wave of activity, and they keep doing that together, depending on again like the level of excitation and connection between the neurons. So that was kind of our model to have this like synchronous wave of activity, and so you can also just see it on this 2D plot, like with the time on the horizontal axis and the activity level, so burst again of activity. And what we found is quite striking is that when you add actually on top of that periodic stimulation that kind of mimic the deep brain stimulation current that we have in the patient, so this kind of regular kind of pulses, depending on the interval between the pulses, the frequency, we see that we can actually abolish those oscillations. And that's what you see here in the middle graph. The oscillations no longer really happen. And so to add a little more on this, the important thing is that, as I kind of mentioned, the neurons are a little deaf in a sense when they are in those big bursts of activity. And so when you add, for example, an initial like incoming signal, they don't really respond to it. They stay, you know, in this very oscillatory mode. And what deep brain stimulation allows, in a sense, is that by abolishing those oscillations, while some heterogeneity and diversity, the signal can emerge again, and that's what you see in this middle panel here. Like, and that's a way of like seeing that, you know, you can retrieve more information kind of flow under the therapy that you cannot get in the Parkinsonian condition. So you can decode somehow the incoming signal a bit better. And so, well, we kind of tried to push that idea a bit further by going back to the clinic and applying it to the Parkinsonian patients. And so the idea here is that, again, deep brain stimulation is very efficient, but it can also always be improved in some ways. And the stimulation parameters can be tuned depending on like the state of the patient, or whether or not he really needs at this precise moment to actually be under like a continuous stimulation. And so what we try to see is that so we tried to see if we could decode more or less well the movements that were actually like done by the patients when the stimulation was on and off, and like the overall aim at the end would be like to see if we adjust the TBS parameter kind of optimally, this also translates into an optimal kind of decoding of the movement that we could then, you know, anticipate and predict like to adjust the parameters in real time. And so we found indeed that there was some improvement when the stimulation was turned on in the way that we could decode the movement that were executed by the patients. I'm not sure how much time I have left. 5 minutes? Yeah? Okay, cool. And so it's kind of, there is a big leap, as you can tell from the mathematical kind of model that I showed you just before, and this very preliminary kind of clinical trial that I was involved in, and that's also kind of the beauty, I think, of the research that I've been able to do, and that was also possible because of the collaborations between like more fundamental labs and clinicians, in our case, like back in Paris. So, and that's again something very common to the research, and especially again here in Boston. That's very nice. And so, just to finish up, maybe with the kind of last story, and again a few words on that. And so, I'm kind of now interested in understanding how hormones can impact our kind of daily, moment-to-moment behavior, and especially like we've been thinking about hormones as having impact on a relatively slow time scale, like days or like weeks even, like depending on the hormones we're thinking about. But in the case of, for example, corticosteroid, which is usually called like this kind of stress hormone, what we know is that there are also some very fast fluctuations that happen during the day. And this is an example of the concentration of corticosteroid that has been measured like at 20 minutes kind of interval time point in a rat, and you can definitely see like very peaky like pulses of corticosteroid release. And how does that relate to behavior is still very much unclear. And so that's kind of the project that I would like to study a bit more. We have a few clues from very old observation, and that's also what's kind of nice with the science, is that we can read very old papers back in the 1970s where the histograms were kind of made, handmade almost, like as you can see in this graph, and related to now like a more precise description of the behavior. And so I've been doing this also by aligning the behavior to neuronal activity. And so to give you a sense of the tools also that we have now to record neuronal activity in rodents in this case. So this is what I implant typically in a mouse. So it's a very tiny probe. If you look at it doesn't really, yeah, it disappears. But so the shank is really like the very small tip that you almost don't see, and that's where the recording sites that allows you to record each single neuron are. And so a typical trace that you get from recording with this type of probe are those like dark trays, where each peak that you see here corresponds to an electrical signal that is actually emitted by a neuron. And if you kind of transform and reorder a little bit like the activity of those neurons such that you produce this big map of neurons that have been recorded like simultaneously, so we can actually access quite many neurons at the same time using those probes. Well, you can definitely see some patterns emerging in the activity, right? Like you see some neurons that fire in kind of bursts, and those bursts actually correspond to episodes where the mouse is actually eating, for example, or grooming. So you can delineate a little bit more what's going on in the brain together with the behavior. So this project is still like very preliminary, so just sharing a few ideas here. And so yeah, so I think I'll finish also by thanking the people that I've been working with, so my PhD advisors back in Paris, and also actually at Brandeis University. So Laurent Delons is the Collège de France, Jonathan Fouboul has moved actually to Brandeis University while I was starting my PhD, and now I'm doing my postdoc at the Harvard Medical School with Professor Tata. So, and thank you again, and now feel free for questions.